So Anto Trimarsanto is also a long-time collaborator. He used to be based in Jakarta, Indonesia, and now he's at Menzies in Darwin, and he's been doing quite a lot of work on various aspects of PV genetic epidemiology, and in particular, he's published a platform for distinguishing the origin of PV parasites. So he can talk a bit about that. Thank you, Anto.
Thank you. My name is Anto. I'm currently based in Menzies School of Research in Australia. I'm working under Dr. Sarah Auburn. First of all, I would like to thank the GenRe-Mekong projects and my good friends Oliver for inviting me to Bangkok. It's always a pleasure to be here. My last time in Bangkok is, I think, 7 years ago, so it's a long time ago. So yeah. So today I'm going to speak about how we predict the geographical origins of vivax infections. So, but it's more on global view. So probably not as fine map as the GenRe-Mekong things, but this is also usable because as you know that the imported cases of malaria confound local controls efforts and enhance the risk of the drug-resistant spread and outbreaks. So I'm not going to reiterate the things about the important cases of imported malaria, but what I want to emphasize is that vivax has the ability to form dormant liver stages and all the asymptomatic cases like the low parasitemia. Hence, reliance on the patient travel history might not be that accurate. So we want to be able to distinguish the local and the imported cases of vivax using a much more honest stuff, which is the genetics of the parasites itself. So there are several previous studies that has been looking for the marker of the geography for vivax. There are 3 studies that are using the non-recombining mitochondria and apicoplast genomes, but the resolution is not that great. There are also studies using the SNPs in the nuclear genome, notably by the broad teams. And this is quite remarkable because they actually only work with 13 genomes datasets compared to the latest days now that we have more than 1,000 whole genome sequence data. Apart from with those limitations, they're still able to develop 40 or to find 42 SNPs that can be used to facilitate fingerprinting and also some kind of the geographic assignment by using a principal component analysis. But using component analysis is a bit subjective because if you see here one of the samples here, the red one, it's clearly it's samples that sampled in this country, but the genetics is actually belongs to the, if I'm not mistaken, is Indonesians. So yeah, it's subjectively right. But what happened if the samples belongs to these overlapping regions? We don't really know, and it's this subjectivity will confound the actual predictions. And also that Sanger Institute has developed assay for 38 of those 42 SNPs. I guess the rest of the SNPs, the full one, is probably difficult to have a primer designs around those position. So the VivaxGEN-GEO objective is to develop the most optimized way of genotyping tools for country level. So this is also important. We are looking at the country level as objective as possible, but still leave room for some subjectivity interpretations, as user-friendly as possible as well. And it's usable with the current and future genotyping technologies, and also amenable to SNP updates, like if you want to add new countries or probably different years if there's a genetic shift over time, and also that it can work together with other SNP sets. So we want to preserve the continuity of all those SNP sets that have been genotyped, so that those data won't be just being unusable anymore. So our work has been published in the Nature Communication Biology 2 years ago. If you want to see the details of the algorithm and how we actually do the stuff. So in this talk, I will just pass along with the contents of what the classifiers that we use, what selections that we employ for finding the SNPs, the performance validations of those SNPs, the online tools that we also develop, and also the assay designs for doing the actual genotyping of the samples. And also some future works that we've been doing for this project. So the classifiers that we use is a modified Bernoulli naive Bayes. I'm sorry if it's a bit technical. This is the thing that we modify so that it can handle biallelic data and provide so that we can have the reasons that we use naive Bayes is that because it provides information on the confidence of the predictions, and also it is able to process missing data, which might come from the low quality samples, and also heterozygote calls, which usually result from the polygonal infections. So this is the thing about 5X. You usually have more heterozygote calls compared to falciparum. So I don't know why the thing in GenRe-Mekong actually has less heterozygote calls compared to the falciparum one. And we also want so that the classifiers are amenable to SNP set additions, as I said before, new countries, genetic shift over time, and others. So the datasets that we use came from our partners from the 21 countries. So we are very grateful for their cooperation and collaborations. And if you look at the whole genome sequence data, it is really possible to identify the infections on the country level by looking at the NG, the global NG tree. And now the question is, because nobody will going to do whole genome sequence things if it's the question is just when or where the infections come from. It's too expensive. So we would like to find a SNP set with a minimum number of SNPs that could differentiate at the country levels. And we employ 3 models to find those best SNP sets. The first one is the usual decision tree. And the second and third is I call it a hierarchical FST. So it's like a decision tree, but with a guided populations tree to help finding the SNPs based on the FST of the closest branch. So what FST means is that it's an index that if we have 2 populations with more or less the same allele frequency between each other, the FST will be low. But if the allele frequency between 2 different populations is very different, then we can assign high FST, and that we can separate those 2 populations easily by that difference in allele frequency. So we end up with of course 3 set of SNPs, the GEO33, the GEO50, and the GEO55, because it contains 33 SNPs, 50 SNPs, and 55 SNPs respectively. And when we look at the performance of those SNPs by using the stratified cross validation, so the cross validation is essentially we divided those training set into say 10 different groups. We use 9 of the groups as the training set and 1 of the group as the validation set, and we do it iteratively, randomly, so that we can have a better picture of how good the SNP sets for doing the predictions. So using the Matthew correlation coefficients, MCC, the SNP sets performs better or actually good enough in identifying the country of origins. But if you look at some regions like the Vietnam and Cambodia, it's actually it looks low, but I'll come to that. And for the rest of the regions, it's actually I'm quite surprised that even the broad barcodes can do this stuff, even though they only work with the 13 genomes dataset. So it shows how remarkable those teams at finding those SNP set. We also do the missing simulations because, as you know, not every samples can be guaranteed to have all markers successfully genotype. And this is because some samples might have a low quality. Also that it's possible that there's some variations at the primer binding sites or regions that we use for designing the primers. So using that simulations, we can be certain that 10 or 20% of missing data, the prediction is still accurate enough. And even for up to 30% missing data, both the algorithm and the SNP sets can still works wonderful, except in some regions where the data might not be as rich as we would like. And we also perform the assessment using the independent validation dataset. So this is another dataset that we will not use for the training step. And if you look at C, if it's in diagonal, so that means the predictions is accurate to the point of the country, and this is why the Vietnam and the Cambodia looks worse in the previous cross validations evaluations, just because some of the samples in Vietnam will be tagged as the Cambodia and vice versa. So this is the problem with this I think the barcodes, because with the shared borders regions, because of people movement and everything else, it's a bit difficult to pinpoint where the sample comes from when it's already like happening in that borders regions anyway. So the limitations of this methods, of course, if the country is not in the part of the training set, we cannot predict that country at all. It won't even it won't even show up in our algorithm. There's some low confidence for some regions, such as the West Asia between the Afghanistan, Iran, and India. I guess northern India, where I think people movement is already a bit loose in that regions anyway. But we are trying to refine those regions. And also in the shared borders examples like the Cambodia and Vietnam situation. So we developed the online tools that's in that URL. So it only requires the actual barcode to be put, and we can choose whether we want to predict on the country itself or on the region. So this is like whether it's from the West Southeast Asia or from the Southern Africa, Southern America, or those big regions. And once we put the barcode, we can just run the classifiers, and it will come up with at much as 3 predictions if it's possible. So for examples, for this Colombia samples, because even the first predictions is actually have a high confidence levels. One is like almost 100% confidence in the algorithm. Then there will be no second and third predictions. But for samples like Myanmar here, the algorithm predicts that the likelihood of the samples coming from Myanmar is 0.83 to 80%, and the rest is probably it's from Thailand or from Bhutan. The algorithm also detects if there's a mismatch barcode, like if we accidentally put, say, the last base here is T, while the algorithm expects either A or T. If we put it T and the algorithm actually expect A or G, then it will complain and mask that base and then show what's how much that is actually masking, and even if it's not in the correct length, it also complain. So this is based on the country, and you can also get if you choose the right the correct model, then you can get even the big regions, and the big regions will always be much accurate than the country level itself. So now it's coming to the assay. So we have the SNP sets, we have the algorithm, but if you don't have the assay to actually genotype the samples, those things can just go to the bin. So fortunately that we've been working with the team from the University of Antwerp in Belgium to design the GEO 33 panels. I personally wish that they have chosen the GEO 50 or 55, but that's what it is for now. And this is just a set of primers that can amplify short DNA regions using the Illumina platform. And they also add another 40 SNPs specific for Vietnam. And this shows the ability of the GEO panels that it can be incorporated into any barcodes to enhance whatever they need to do with their own barcodes. So they already published the assay and some of the results in this journal, and it's very funny because this journal actually published first before our own publications, just because the review process actually takes longer at our paper. So we also would like to have the assay in the ONT platform. Why? Because the biggest selling point with ONT is not because of the portability. I mean, the sequencers is portable, but the rest of the things like the PCR machine, the centrifuge, is not that portable anymore. So you still have need to set up for doing the sequencing anyway. But the biggest selling point is that the flow cells can be reused. So if you know the mechanism with the Illumina instruments, you have the flow cells, but you can only use it once. So if you just run for 5 samples, 10 samples, or even 96 samples, once you use the flow cells, and that's it, you cannot reuse it anymore. But with the ONT, the possibility of reusing the flow cells actually is very suitable for low throughput sequencing projects, so it can be easily used in a very low transmission setting or regions. So we are adopting the Illumina assay to be used in the mean ion assay, and it's been led by Angela Rumaseps, one of our wonderful PhD students under Dr. Sarah Orban's team. And the concordance with the whole genome sequence data is roughly about 90 and 95%, and we know that mean ions has this error rate problems, and we try to address this with some stuff that we are going to do, as I'm going to say in next stuff. So with the depth itself, it's actually looking good that the mean ion assay is roughly comparable with the Illumina assay, as that it can produce depth high enough for our purposes. So amending SNP, how easy is it to amend SNP to our GEO panels? So I'm just using an example from the Indonesian side. So we just recently have whole genome sequence data from Sumatra, Indonesia. And our previous training sets only use Papua, Indonesia, which is in the eastern part of Indonesia. So the Papua is here, the Sumatra is here, and if we look at the neighbor joining tree on those regions, it's very easily can be seen that the Sumatra, Indonesia is actually closely related to Malaysia. So if we use the current SNP set and current training, all the Sumatra samples will be identified as the Malaysia samples, and we don't want to do that. So we can then select candidate SNPs just by using or finding some SNPs that have a high FST between Sumatra and Malaysia, and we can then incorporate that candidate SNPs for each candidate SNP and perform cross-validation with the merged SNP sets, and also validate with the independent validation data, just to make sure that the additions of this edit SNPs won't degrade the previous performance. And also, if we have several candidates, then we can design the assay easily, because sometimes, even if we want to be able to genotype in that regions, it might be impossible to design primers around that cell. Probably it can interfere with the established SNP, the established primers for other SNPs. And for adding a lot more new countries, we also have luckily we have Ludwig Hoon, one of our new postdoc in Dr. Sarah Auburn's team. So it's going to amend the panels for some new countries using the latest public whole genome sequence data, and we want to refine the West Asia and the South America regions, because some of the new samples actually came from that regions. And if you're wondering when I'm saying about what population tree is, this is what population tree looks like. So it's, instead of samples, it's showing you the relationship between countries. And what happened with the microhaplotype panels? We also want to see whether the microhaplotype panels, as it is by the SNP, can be used for global predictions, and it actually does perform quite well, considering that we actually don't design it for geographical markers. So for the future works, we are going to refine the classifiers to work with the actual microhap data. So instead of looking at the SNP, looking at the actual microhaps for both the VivaxGEN's microhaplotype panel and also for the geo assay, because when we perform the genotypings, we actually produce amplicons that can be considered as a simple microhaplotype. We also want to update the online tools to accept VCF files and the raw FASTQ files, so that people do not have to perform their variant callings independently. But for bulk cases, we still hope that they perform the variant callings locally before submitting into our web tools. And also we want to reclassify shared border samples from the respective main countries. So examples, we have country X and then country Y sharing borders. We might have another classification for country X plus Y, which means, okay, this is part of the borders that we don't have to deal, because it's already done deal in that border regions anyway. So I would like to acknowledge all the people that's been helpful in this odyssey. Rick Price, Professor Rick Price and Dr. Sarah Auburn's, who allow me to perform this kind of project. My former colleagues in Ekman Institute in Jakarta, Indonesia, especially Dr. Rintis Novianti and her malaria pathogenesis team, who's generating most of the data from the Indonesian sites, and also the Wellcome Trust Sanger's team and the malaria gen people, especially Richard Person, who did the original whole genome data variant callings for the global populations, and also Oliver is there as well, and the late Dr. Professor Dominic Kwiatkowski, and all the founders, and also our lovely collaborators under the VivaxGEN's umbrella. Thank you very much.
Thank you very much for that wonderful work, Anto. I think we have time for one question for Anto, if somebody has one. So I actually have one. So
you are in the paper.
No, but did you consider, because right now the web app accepts the barcode,
Yeah.
but perhaps later on, let's say if a control program works with the lab that has a sequencer, they may just be getting the sequence data and they don't want to derive the barcode. They just want to know the answer. Okay. Or the raw FASTQ file. Okay. So, do you have any feedback as to whether, whether there are groups ready to use that? Okay.
Beginning of next year? This is already almost next year, but yeah, I promise once this actually is quite easy because we already have the pipelines for doing the raw FASTQ files anyway that you already use. So we just need to integrate it into our web tools, and it should be doable within actually 2 weeks if I have time.
Okay, great. Sarah.
Yep, thanks. Just to comment on that question about groups who are ready to use it, that the Antwerp team and our Rosanna's team are actually already using it. So and hopefully it might be useful for places like China and other sites in the future.